Hello, today we're going to talk about energy in cells, how it's made and how it's used. We've already talked about photosynthesis and respiration a little bit, but we're going to dive deeper into these processes today. Remember that organisms that can do photosynthesis have chloroplasts and or chlorophyll, and you see a few chloroplasts here in the plant cells on your screen. These photosynthetic organisms are capable of converting sunlight into glucose, and that glucose, all organisms, including those photosynthetic ones, can use to break down during cellular respiration for energy. So, energy is used in cells for a variety of reasons. These are just a few examples. Cells need energy to make new molecules like proteins and nucleic acids. Energy is used to build and repair membranes and other organelles once they've been damaged. And a lot of energy goes into maintaining homeostasis or balance in cells by moving molecules back and forth across membranes. Remember that homeostasis is really important for life because it allows cells and organisms to maintain their internal environment regardless of what's going on outside of the cell or organism. The energy currency used by cells is called adenosine triphosphate or ATP, and tri means three. So if we look at this ATP molecule here, we can see it has three phosphates, and these three phosphates are attached to a ribose sugar and an adenine base. And when we have a sugar, base, and phosphate, then we have a nucleotide. So ATP is a modified RNA nucleotide that's modified for carrying energy in cells. This ATP is going to be made in the mitochondria, which we can see here. And the mitochondria has this folded inner membrane. And this is folded to increase the surface area to fit more enzymes on it that are capable of making ATP. The more surface area, the more ATP we can make. So here is another picture of the mitochondria, and this was taken by an electron microscope, and you can see this folded inner membrane here again. In order to release the energy in a molecule, bonds must be broken, and to store energy, bonds must be made. So to release the energy in ATP, we're going to cut the bond between these last two phosphates, and we're going to do this using hydrolysis, which you've heard of before. Hydro for water and lys for to cut. So we're going to take a water molecule and insert it between these two phosphates and break this molecule apart. That's going to release energy, but we're no longer going to have ATP anymore because we don't have three phosphates. Now we're going to have ADP or adenosine diphosphate. And di means two because we only have two phosphates. Now this is a very low energy molecule. And so in order to get back to our ATP state, we're going to have to make a bond to store that energy. All right. So if we look at how this energy is going to be supplied to make the ATP, we're going to be breaking down carbohydrates, proteins, and lipids. And that's going to allow us to reattach this phosphate to ADP to get ATP. And this ATP energy is going to be used for creating new molecules, getting toxins out of the body and the cell, making heat, moving ions or other molecules, and for animals, muscle contraction. So I have a little demonstration I'll do to try to help you remember this. If I've got these two molecules here, let's say this is my phosphate and this is my ADP, and I need to store energy, then I'm going to make a bond between them. So I'm going to use my rubber band here, and I'm going to make a bond so that now these two molecules are held together. Now, if I cut this bond, then I should release energy. So let's just see if we can see this energy be released. And hopefully you saw the rubber band fly across the room. So that tells us that when we break bonds, energy is going to be released. And in this demonstration, that energy goes into moving that rubber band. ATP is constantly being used in the body as an energy source. So this last phosphate is always being broken off by adding a water molecule. And then we no longer have ATP, we're going to have ADP. So there's no energy in the ADP molecule. Now, in the mitochondria, ADP goes through a process called phosphorylation. I don't care that you know this term, but I want you to understand what's happening. We're taking this phosphate and attaching it to an ADP to generate ATP that can then be used for energy. So this goes around in a circle, and we call this the ATP ADP cycle. Now, it's important to understand that ATP cannot be stored for later use. It's used about as quickly as it is made. In order to attach phosphates to ADP to get ATP, we need an energy source. And most life forms are going to depend on photosynthesis as that energy source. That doesn't mean that all organisms do photosynthesis. That just means that we all depend on the products made by photosynthesis. So, this is a process where plants or other organisms with chlorophyll are going to join together carbon dioxide and water by using the energy in sunlight. So they're going to convert that sunlight energy into carbohydrates or sugars like glucose, which are stored in the leaf and may be sent down to the roots. As a byproduct, photosynthesis is going to produce oxygen. Well, we and other organisms can use this oxygen during respiration to break down that carbohydrate, that glucose. Plants do respiration as well. So either this oxygen is going to be released or it's going to be used again by the plant cell to break down the glucose that they've made to support their own life processes and give them energy to make that ATP molecule. Here are just some examples of organisms that can use photosynthesis. We have plants, from your mosses to your big trees, algae, which you can see here in this water, and some protists, like this euglena here. A protist is usually single celled, can be multicellular, but it is a eukaryote. You can see it does have a nucleus here. And this particular protist, the euglena, is capable of doing photosynthesis in the same way that all of these are, because they have those chloroplasts. There are some bacteria that can carry out photosynthesis, but they do not have chloroplasts. They just have the pigment chlorophyll. Here's your photosynthesis equation, and it has been balanced for you. It's the sixes in front of the oxygen, water, and carbon dioxide. But we're just worried about the general formula. So we can see our reactants, carbon dioxide and water, which are put together using the energy in light to create our products, sugar in the form of glucose and oxygen. Now remember that we do not include light in the equation because it is not made of atoms. It can't be rearranged. It's just an energy source, and so it goes over the arrow. Most photosynthetic organisms perform photosynthesis in their chloroplast. We see a chloroplast here in this diagram of a plant cell. We can see a much larger chloroplast here. So we have an outer and inner membrane, and then inside of that inner membrane, we have these stacks called thylakoids. And this is where the pigments are going to be that absorb that light energy and convert that energy into the chemical energy in the form of glucose. Now, the main pigment is going to be chlorophyll, and chlorophyll is green. This is why plants look green when we see them. This chlorophyll reflects green wavelengths back to our eyes, and that's what we pick up. There are other pigments as well in the chloroplast that can do photosynthesis, but they are less common. However, we start to see those this time of year when this chlorophyll breaks down. And that's why the leaves turn red and orange and yellow in the fall. It's these other pigments that we are seeing that do photosynthesis under different wavelengths. Photosynthesis is a two-step process with two different sets of reactions. The first set is light dependent and it requires sunlight. The second set of reactions is light independent and it does not require sunlight. We also call these the dark reactions or the Calvin cycle. Now, don't be confused. Saying that they are light independent does not mean that they can't happen in sunlight. They can, but they do not require sunlight to generate the energy to do these reactions. So the light-dependent reactions happen first in photosynthesis, and this is where chlorophyll is going to absorb energy from the sun to create these energy carriers, ATP and NADPH. And ATP and NADPH are going to be used later in the light-independent reactions. So we'll just take a little glimpse at how this works. We have water coming into the chloroplast, and this is in the thylakoid. So we're on the membrane of one of those thylakoid stacks that we saw. An electron is going to be ripped off of this water and taken into this first photosystem. Light comes in and gets this electron all excited, so it starts bouncing around and it can't be contained anymore, so it starts running down this membrane of the thylakoid. As it does, it changes the shape of this enzyme here, pumps hydrogen across this membrane. So it's going to start building up. That's going to come back into play. This electron is going to keep going until it gets to this next photosystem. And now it's lost most of its energy, so it's going to be contained in this photosystem. Now this light energy is going to come in again and get this electron all excited. And once it does, it's going to leave this photosystem and go through these two little enzymes here, which are going to create that NADPH. Okay, so there's our NADPH coming out of our light-dependent reactions. Now remember, I told you that hydrogen was being pumped across this membrane, so we're building up a concentration of this hydrogen. And remember that things tend to diffuse from a high to a low concentration. So this hydrogen is going to diffuse through this enzyme called ATP synthase. And this is the enzyme that's going to attach that phosphate to ADP in photosynthesis. So here we're getting our ATP. And that's where our NADPH and our ATP are going to come from in the light-dependent reactions. I don't expect you to know all that. I just want you to know that NADPH and ATP are made during these light-dependent reactions. I just think it's cool to know how that happens. The ATP and NADPH molecules that were made during the light-dependent reactions are now going to be used in the light-independent reactions, which are also called the Calvin cycle. Now, we do not need sunlight during the Calvin cycle because we're getting our energy from this ATP and this NADPH. So the Calvin cycle is where glucose is actually going to be made by joining together carbon dioxide and water. Now, I'm not going to go into this in too much detail. As you can see, there's a lot of steps, but I do want to draw your attention to a couple of things. We have this starter molecule called RuBP, okay? And this is where the ATP is going to come in, and we're going to attach that carbon dioxide to the starter molecules using Rubisco. Rubisco is an enzyme you've heard me mention before. It's the most plentiful enzyme in the world because all organisms that do photosynthesis have it. So we're going to be using this carbon dioxide to make these molecules that are then going to have more ATP and NADPH put into them until we get these 3 carbon molecules that can be attached together to create glucose. Now, we also need to remake our starter molecule from this as well. So big things I want you to take away: ATP is added, NADPH is added, and our molecules, glucose, are going to come out here. So photosynthesis is the process that's going to convert that energy in sunlight into chemical energy like glucose. And that glucose is going to have energy stored in its chemical bonds that we and other organisms can then take in as food to create ATP energy during the process of cellular respiration. Now, when you eat a lot of sugar, that sugar, if it's not used, can be turned into fat. And when you don't have enough energy in sugars, then you can take this fat and convert it back into sugar that your body can then use for cellular respiration. So this process is going to release energy from sugars and fats. The most important thing that it's going to use is glucose. And glucose is going to be converted into ATP by the mitochondria. So if we look at our equation, we have our carbohydrates going in, our nutrients, and our oxygen. And then we are converting this into energy and releasing carbon dioxide and water. So notice that the things going in to cellular respiration are coming out of photosynthesis. And the things that are coming out of cellular respiration go into photosynthesis. So these equations are opposite of one another. Cellular respiration is a 3-part process. You can see the 3 parts here, and we're going to do a general overview of this. We're not going to get into all this detail, but there are some important things that you need to realize that are going on here. So we'll start with glycolysis, glyco for sugar and lyse for to cut. So we're going to be cutting sugar here. This occurs in the cytoplasm with or without oxygen present. It doesn't matter. It's not used in glycolysis. So our 6-carbon glucose comes in, and it's going to be cut in half to give us 2 3-carbon molecules called pyruvate. Now, in order to do this, there is an energy investment stage where we're going to use ATP. So 2 ATP are going to come in, but we're going to get 4 ATP out in the energy harvesting stage. We get 2 here and we get 2 here for 4. So it's the same thing if we think about money. If we use $2 in an enterprise and we get $4 out, then we have a net gain of $2. In this case, we have ATP. We've used 2, we got 4, so we have a net gain of 2 in glycolysis. Now, once we have this 3-carbon pyruvate molecule, it's going to go into the mitochondria and go through the Krebs cycle. Now, the Krebs cycle is going to generate 1 ATP for each pyruvate, and we have 2 of them. So the first one comes in and we get 1 ATP. The second one comes in and we get another ATP for 2 ATP. We're also creating this other energy carrier called NADH. And this NADH is going to be used in the electron transport chain, which is in the mitochondria's membrane. So this NADH is giving energy to electrons so that they can run through this chain, kind of like our sunlight did in photosynthesis. As these electrons run down this chain, they're going to be used to concentrate hydrogen on this side of the membrane, which is going to go through that ATP synthase enzyme that we looked at earlier, and we're going to generate ATP from that. Most of your ATP is going to come from this electron transport chain. You get 34 ATP for each glucose molecule just in this electron transport chain. So when we think about it and you burn a single glucose molecule, you're getting 38 ATP out of a single glucose. So that's pretty efficient as far as energy use is going to go. Now we have 2 kinds of respiration, aerobic and anaerobic. Aerobic is a process that requires oxygen. That's what we're going to find in the Krebs cycle and in the mitochondria. So the Krebs cycle is an aerobic process. Anaerobic types of respiration do not require oxygen. So glycolysis would be an example of an anaerobic type of respiration. Another example of anaerobic respiration is fermentation. This is a process that allows cells to get energy in the absence of oxygen, and there are 2 important types: lactic acid fermentation and alcohol fermentation. And in some shape or another, humans are going to exploit these 2 types of fermentation. And we can see here some ways I want you to remember fermentation. These are wine casks fermenting wine. And this is a brewery where they are making beer by fermenting mash inside of these big barrels. Fermentation always starts with glycolysis. We are cutting sugar. So that glucose with 6 carbons is going to cross the cell membrane and get into the cytoplasm, and it's going to immediately be cut in half into 2 3-carbon molecules called pyruvate. Now, this has another function as well. If that glucose is broken in half, then you're going to steadily keep more glucose outside of the cell than inside. That way, glucose is going to constantly be wanting to diffuse into the cell. Now, during glycolysis, again, we're going to use 2 ATP, but we're going to get 4 ATP back out. With the 2 that we used, plus 2 more, so we have a net gain of 2. And this is all occurring in the cytoplasm. So there were 2 types of fermentation that we needed to look at, and we'll look at lactic acid fermentation first. This is where we are going to take that pyruvic acid through an enzyme chain that's going to convert it into lactic acid. Most cells can't use lactic acid and they get rid of it as waste. This occurs during your muscle cells when you're going through strenuous exercise. If your muscles aren't getting enough oxygen to continue to go through aerobic respiration, then they're going to switch to going through lactic acid fermentation. That lactic acid is this milky white substance that sits on top of the muscles and makes them sore. So if you know that you are going to be sore after a workout, here's a little hint for you athletes. A big glass of apple juice will cause this lactic acid to break down faster, and it will decrease the amount of time that you stay sore after a workout. Now, we humans have also exploited this lactic acid fermentation in other organisms to make things like yogurt, cheese, and other dairy products. So, again, we're starting with glycolysis, breaking that glucose into two pyruvate molecules, taking it through this enzyme chain, and getting lactic acid or lactate out of it. Ethanol fermentation is another type of fermentation that humans benefit from, but we can't do this type of fermentation in our bodies like we could lactic acid fermentation. Now, it's going to start in the same way. We have our glucose molecule that's going to go through glycolysis and get broken into our 2,3 carbon pyruvate molecules. And those pyruvate molecules are sent down an electron transport chain to create ethanol. And no, this is not English, but it's close enough and it gets the point across. Now, most organisms that make this ethanol can't use it, and so they excrete it as waste. And we can distill this ethanol to make things like biodiesel, beer, and wine. And you can see here an old moonshine still, and we're from Wilkes County, so you've probably seen these a lot on display out in town and around places. But most of the time, we're using bacteria to do this ethanol fermentation. However, yeast is capable of doing this as well, and yeast is a eukaryote, it's not a bacteria. So we use yeast in making beer and we use yeast in making bread. Bread rises because of ethanol fermentation, and those little bubbles that you see inside of the bread are a result of this process. Here are my resources, and I hope you learned a little something about how energy is made and used in organisms.